the Land Institute, we seek environmentally friendly methods to manage insects and pathogens in perennial grain crops. Plants have evolved many natural chemicals to defend themselves and to attract pollinators.
Identifying these natural chemicals can help us to discover what organic compounds each plant produces.
To study these chemicals, we use gas chromatography-coupled mass spectrometry, or GCMS, to quantify their molecular structures. But identifying chemicals from GCMS data is a time-intensive process. To address this bottleneck, we have developed a new package for R called uafR. Hi, my name is Chase Stratton. I led the development of uafR.
My name is Yvonne Thompson, and I am a research associate at the Land Institute, and I was involved in the ideal conception and beta coding of this R package.
Hi, I'm Ebony Morell, lead scientist in crop protection ecology at the Land Institute. My role was to help troubleshoot the R package and to help test its efficacy compared to traditional methods of GCMS chemical ID, and also co-writing the manuscript.
I'm Connie Lozio, data scientist at UGLAP. My contribution to the design of the uafR package consisted in sharing my experience building R packages.
I'm Dr. Rob Morrison. I'm a research entomologist with the USDA Agricultural Research Service in Manhattan, Kansas.
My name is Philip Hansen, and I was a visiting scholar with the Crop Protection Ecology Group at the Land Institute during the development of this package.
The original motivation for uafR was to be able to interpret the volatile samples collected during our perennial biculture greenhouse trials with TLI crops.
As someone who regularly runs chemical ecology data, I found that the rate-limiting step is actually the data analysis part.
During the process of learning how to code for GCMS data analysis, I was stunned by the inefficiencies in the workflow and decided to start developing a new, easier pipeline in R.
When we realized that accessible R packages for GCMS data were hard to come by, we were intrigued to develop the code we had written into something that would be available to anyone that might find this useful.
If we could actually speed that process, we can speed discovery, interpretation of data.
However, it quickly became apparent that the code we had been developing was way too useful to keep in-house.
uafR uses online published chemical data from PubChem and other sources, as well as the mass and configuration of compounds detected by the GCMS to identify chemical compounds in multiple samples of a research data set. uafR can also classify those compounds into categories, standardize the abundances of the compounds using an internal or external standard, and compile the identified compounds into a data set that's ready for statistical analyses.
It makes complex cheminformatics workflows accessible to anyone with basic experience in R.
Anyone with GCMS data needing to be analyzed can take advantage of this open source, user-friendly R package.
This package will not only make an entire experiment worth of mass spectrometry data available for publication quality analysis in a couple of hours, but take subjectivity out of the equation. It's faster, more accurate, and free.